Hello guys, I am Shiva. Welcome to Minerva. So in this class we will discuss the class 10 ICC class 10 first chapter of biology. Okay. So what actually in class 10 biology first chapter, that is cell cycle, cell division and chromosome. So let me tell you that in this section we will discuss all the chapters, okay, all the chapters of biology. First I am starting the first chapter, and regularly we will update all those videos and all those chapters they are included. Okay, so let's start the first chapter, which is cell structure. First understand actually the cell structure. Okay, we will understand cell division. Before that, understand the cell structure, what actually cells are. First, let me tell you, there are two types of cells we can see on the basis of plant or animal cell. Okay, and if I tell you that the things which is present inside the cell, like cell organelles, like nucleus and all those organelles, like mitochondria, ribosome, on this basis, if I categorize of these cells, we can say that there are two types of cell can be present, or two types of cell can be seen. One is prokaryotic and another is eukaryotic. Okay, prokaryotic is all those type of cells which is already primitive type, means those type of cells were existed in that evolve or that produce in the early time of in this earth. Okay, so like in the case of primitive cell or prokaryotic cell, this type of cells are containing the nucleus which is not developed as much as like eukaryotic cells. Okay, like if I tell you that the nucleus, which is the structure, let me show you the nucleus. If this is the nucleus present in the eukaryotic cell, suppose this is a eukaryotic cell and this is the nucleus of the eukaryotic cell. So this nucleus is proper developed, means this nucleus is containing the nuclear membrane and the nucleolus and all the things which is present, like chromosomes which is present inside the nucleus. So in the case of eukaryotic cell, this is proper developed, means the nuclear membrane is here present. Nuclear membrane is present means the chromosome or the scattered DNA which is present inside the nucleus, that is totally bounded inside the nucleus. And in the case of prokaryotic, they are genetic materials. Actually, let me tell you, this DNA or the chromosome, inside the chromosome there is DNA, means the chromosome contains with DNA actually. Okay, so if you unfold this chromosome, you will find that this is containing actually DNA. So this genetic material is bounded with the cell or the nuclear membrane. In this case, this is the eukaryotic cell. But in the case of prokaryotic cell, just their nuclear membrane is absent. Okay, their nucleus membrane is absent. That means the genetic material is scattered in the cytoplasm. Okay, let me tell you what is cytoplasm. Cytoplasm is the present inside the cell, or inside the cell, the jelly-like substances where all the cellular organelles are floated. That things we call the cytoplasm. Okay, so this area which is present around the nucleus or around the genetic material or nucleoli actually, so this scattered things is the cytoplasm. Okay, jelly-like substances, and in these jelly-like substances here all the organelles are floated. Organelles means all those things which is present inside the cell. Okay, like that can be mitochondria, ribosome, golgi apparatus, all those things. Okay, which we will understand in the cell chapter. So this is what actually normal cell. This is the prokaryotic cell. If I draw the nuclear membrane, then we will call it a eukaryotic cell. So in the case of cell cycle, what actually happened, let me tell you that in this case just this cell will divide, and because after some of the time this cell will die. Obviously this cell will not stay or alive forever. Okay, so before dying, before the death of this cell, it will produce, or it will divide actually and produce two new cell. Okay, because if your body get cut, then this cut needs to repair. To repair that cut, your body needs to generate new cells, right? Suppose in my arms is in this portion, I got cut. Okay, so after few days it will repair. How it actually repairs? The two side of the cut here, the cells which is present two side of the cut, these cells will divide and it will repair this portion. Okay, it will repair this portion after few times. This time of the cell division actually varies on species to species. Okay, so this is the very important process. Cell cycle means how actually cell is dividing and it is producing new cells. So in this case, cell cycle means one cell before dying it will produce another cells, and that another cell after few times it will produce another cell. So actually these things are happening in a cyclic manner. Okay, how it actually happens? It will not just rapidly divide. Okay, to divide the cell, it needs to go some steps, or it needs to follow some steps. Okay, that steps we will understand. And let me tell you that in our body there are two type of cells present. Okay, two types of cells means on the basis of their genetic material. Genetic materials means on the basis of their, that is, diploid cell or haploid cell. On those basis we can categorize actually. Okay, because haploid cell means the genetic chromosome number that is n, only n means the chromosome in our body. Let me tell you that in our body has 23 pair of chromosome. Okay, our body has 23 pairs of chromosome. So in this 23 pair means 46 number of chromosomes present in our body. Every cell contains, inside the nucleus there is a chromosomes. So number of chromosomes is 46. Okay, how actually we got this 46 number of chromosome? During the fission of gametes, let me tell you, this is very interesting because we got this 46 number of chromosome. Half of the 46 number of chromosome we get from our mother, and half of the 46 number of chromosome we got from our father. Okay, means if we divide it by 23, number of 23 and 23, this 23 number of chromosome came from our mother. Okay, and this number of 23 number of chromosome we got from our father. So after getting this configuration or number of chromosome, 23 from father, 23 from mother, we got this 46 number of chromosome in our body. Okay, so how actually we got this for 23 number of chromosome? Because every cell is containing 46 number of chromosome. In our body, every cell which is containing 46 number of chromosome. But how we actually got this 23 number of chromosome? From our parents. From mother 23, from father 23. How? You have to think about it. This 23 number of chromosome, actually this cell which is this nucleus, or this 23 number of chromosome is actually come from the gamete cell. Okay, so that's what I am trying to tell you that our body, there are two kind of cells are present. One is somatic cell, which we call the body cell, means in our body there is normally somatic cells are most of all. Okay, and there are different type of cells are present, which we call the germ cell or the gametic cell. Okay, so germ cells are containing 23 number of chromosome, and our body cell or somatic cell is containing 46 number of chromosome. So the process of division of germ cell, which is different, and the process of division of our body cell or somatic cell, that is also different way. Okay. So the process of division, which is different, first we will understand the division of our body cell. Okay, how our body cell is actually dividing and it will produce the new cells. For that you have to understand the process which we call the mitosis. So if I tell you that how many types of cell division can be seen in our body, then we will say that there are two kinds of cell division can be seen in our body. First is mitosis cell division, which is happen in our somatic cell, means in our body cells, and meiosis. Another type is meiosis. This is happened in the germ cell or in the gamete production. During the production of gametes, this kind of cells actually produce, or this type of cell division actually happened. Okay. So first we will understand the mitosis, and in this chapter we will also discuss the structure of chromosomes. What actually chromosome? I already told you that inside the cell there is a nucleus, and inside the nucleus there is other substances present like nucleolus and also the chromosomes. So that we will understand how actually the structure of the chromosome. You know, the best time for the study of chromosome, which is the prophase stage, okay, or in the anaphase stage also. So what is this prophase and anaphase stage, that we will understand in here, okay. So first understand the mitosis. In the case of mitosis, this type of cell division, I already told you, this type of cell division happen in our body cell. So from one cell, suppose this number of chromosome, which is 46, it will divide and it will produce two number of cell. After a certain period of time, it will just produce a furrow-like structure, means cleavage-like structure. It will produce. Here, let me show you. That inside, okay. This type of structure it will produce after few time, and it will just divide, or it will just cut off from mid portion, and it will produce two new cell, okay, two new daughter cell. So this method actually we call mitosis, where the number of genetic material, which is 46 number of chromosome, is the mother cell, which will continue, means in the daughter cell that will also 46 number of chromosome, okay. Let me tell you why actually we call this new cell, we call daughter cell. Why we don't call it a son cell? Because this cell has the ability to produce again two new cell. So son or the male can't produce new organisms by itself. So only daughters or the female have the ability to produce new cells. So we call this daughter cell, okay. So in mitosis, inside the cell, suppose this is a cell, inside the cell there is a genetic material and other stuff like mitochondria, Golgi bodies, all those things will present, okay. Nucleus, ribosomes, endoplasmic reticulum, okay. Endoplasmic reticulum, and inside the nucleus there is a genetic material which is present. I already told you. So before dividing this cell, it needs to increase its genetic material and also the cell organelles which is present inside the cell, because it will, after some period of time, it will divide. So before dividing, it needs to increase, or it needs to grow the cell size also. It needs to increase the organelles which is present inside. So for that reason, it goes some steps or some phases, go through some phases which we call the interphase, okay. And after the interphase means the growth section and the synthesization, means the genetic material which will needs to increase, or which needs to double its number. So during that time, it's happening all those things in the interphase period or interphase season time, okay. After that, the nucleus, inside the nucleus, this genetic material or this chromosomes which needs to divide. So this division also occur, and that thing we understood in the phase which we call the karyokinesis. Okay, understand first what actually happened. These things, or inside the thing which is present inside the things which is present inside the cell needs to increase. During that time we call interphase. And after that, what needs to do? This nucleus needs to divide. So during the time of nucleus division, we call it a karyokinesis. And after karyokinesis, or at the same time of the karyokinesis, what actually happened? This things which is already increased or already grow, that needs to also divide, or that division or that thing we call the phase which we call cytokinesis. Okay, so first interphase will occur, means the genetic material and all those organelles which is present inside the cells which needs to increase. So interphase happened. Now what will happen? This karyokinesis means the nucleus will divide. Okay, after the division of nucleus, or at the same time of the division of nucleus, the things which is already increased in the number or size, that needs to divide. So that division we call cytokinesis. Okay, understand the term which we call karyokinesis. Karyokinesis means the karyon means the nucleus. Okay, so karyokinesis means the nuclear division, and cytokinesis, okay, let me tell you that kinesis means also the division. Okay, so cytokinesis means, cyto means the cellular components except the genetic material or the nucleus. Okay, these things we call cytokinesis. So interphase, karyokinesis and cytokinesis, these phases will occur. So if I tell you that how many steps will occur in mitosis division, then the answer is interphase. And this karyokinesis and cytokinesis we combine and we call it together mitotic division. Okay, mitotic phase. Mitotic phase means if I divide this mitosis division into form, first is interphase, means during the time when all the things inside the cell will increase. Okay, after that meiotic phase. So interphase and mitotic phase. Meiotic phase actually happened in the case of meiosis. So mitotic phase. So in mitotic phase, we can categorize this mitotic phase again in two categories, which is cytokinesis and karyokinesis. So mitotic phase can be divided in two phases, which is karyokinesis and cytokinesis. And again, this karyokinesis, again we can categorize, or actually we can divide this phase on four phases. Okay, four sub phases also. Okay, what all those phases are? Because let me tell you that suppose this is a cell which needs to divide. So before division, okay, I will tell you karyokinesis, all those phases. First understand what actually happened. So this is the cell. This cell needs to divide. So for that reason, it will go to go through a steps which we call the interphase. And in interphase, that also can be divided on 3 phases which we call G1 phase or gap 1 phase, and G2 phase or gap 2 phase, and okay, S phase is before the G2 phase. First G1 phase, then S phase which we call the synthesized phase, and then G2 phase means gap 2 phase. Okay, so in these 3 phases we can divide interphase. So first it will increase the genetic content and also the cell organelles, all those things it needs to increase. So for that reason it will go in the interphase, in the phase which we call interphase. So in interphase, which we call, first it will go through a G1 phase. Okay, during the time of G1 phase, what actually happened, few types of protein which will synthesize, means which will produce, and because during the time of cell division it needs lots of protein, okay, because cellular size also increase and the genetic component which also needs to divide or it needs to double. So during that time it needs some proteins. How it actually produce proteins? It will synthesize new proteins. Okay, so during the time of G1 phase it will produce protein and RNA. Okay, during the time of G1 phase, and after the G1 phase it will just double its genetic material. Genetic material means the DNA. The number of DNA which is present inside the cell, it will just double its number by the process of replication, which we call the process is DNA replication. By this method, actually let me tell you that structure of DNA, how it actually looks. This type of structure DNA actually shows. Okay, so during the time of synthesized phase it needs to double its DNA number. Okay, means the genetic content needs to double because it will go to other cell. Okay, so for that reason it will produce more number of genetic content. For that reason it will go through the DNA replication method. Okay, and then after so this time means the time of synthesized phase, so we call this phase is S phase. S phase. So during the time of S phase, what actually happened? Number of DNA or the number of genetic content will double, and after that what actually happened? It will enter the phase which we call G2 phase. G2 phase. Okay, during the time of G2 phase it will stop the synthesizing of DNA. Means in the time of during the time of S phase it will produce or it will synthesize the DNA, but after the finishing of the S phase, whenever it will enter in the G2 phase, it will stop producing the DNA and it will start again, it will continue its production of protein and RNA. So from normal cell to whenever it will start to dividing, okay, whenever it will start to dividing or whenever it will enter in the interphase, during that time some checkpoints are present. Okay, what kind of checkpoints, let me tell you later. Okay, first understand, after G2 phase what actually happened, it will enter the mitotic phase. It will enter in the mitotic phase, and that mitotic phase what actually happened, this cell now it has the genetic content which is double its number. Okay, genetic content or the DNA is doubled in number. Actually number is remain same in this stage also, but the content or the amount is will increase in this phase. Okay, which phase? This phase we call the mitotic phase. So after mitotic phase what actually happened, during the finishing or the end time of the mitotic phase, it will just divide. This time it will just divide and cytokinesis will also occur. Okay, and whenever cytokinesis will occur, it will produce two new cells. It will produce two new cells by cleavage or forming. Means in this during the point of time, this point of time, it will produce this kind of structure. Okay, and the genetic content which is doubled here, it will just divide equally in the both cell. Okay, like this, and it will produce two new cell. So this is the this is how actually one cell will divide and it will produce two new daughter cell. And here in this case, from this position to this position before the mitotic phase we call interphase. Okay, this position we call interphase, and after interphase this period of time we call mitotic phase, which we call mitotic phase. And some cases what actually happened, this after this mitotic phase, few types of cell will start again proliferating. Means it will produce its genetic content, it will just increase its genetic content or all the organelles, and it will again go to another cycle where it will produce again two new cells. But some cases what actually happened, this cell will not divide, I mean it will not enter in the another cell cycle. It will just arrest in a position, means it will enter in G0 cycle. Okay, or G0 cycle, which will sometimes can come and again it will divide and produce new cells. But in some cases what actually happened, this cell will just get arrested and it will not divide. So I told you that some types of cell which will just get arrested in G0 phase. Okay, means that kind of cells after dividing, it will not go another cell cycle and it will not divide again. And few types of cell will go G0 phase, but it will come whenever it will get stimuli, it will enter again in the cell cycle. And few types of cell which will always repeating, just needs, it will divide and it will produce new cells. Okay, so on the basis of this G0 phase, I mean it will stay G0 phase or it will not stay in G0 phase, or it will not enter in G0 phase. On the basis of these things, we can categorize these cells on three bases, on the three category. Okay, if the cell will divide rapidly, like our skin cell, whenever I mean after the maturation, it will just divide and it will produce again two new cells. So this kind of cells we call labile cell. Okay, this types of cell we call labile cell. And if the cell will get arrested in G0 phase, but after getting this certain type of stimuli, it will come again and enter in the cell cycle. So this kind of cell we call stable cell. like our liver cell, kidney cell. Okay, this kind of cell, what actually happened in, suppose in your liver, this portion got cut. Okay, so after certain period of time, this cells which is present in the edge portion, these cells will start dividing and it will generate the cut portion. Okay, again, so whenever the stimuli means the cut, it got cut. Okay, so after it got cut, these cells will divide, otherwise it will not divide. So this method or this process types of cell we call the stable cell. And there are another cell which will never divide again. Okay, like our brain cell or our neurons. These types of cell we call permanent cell. After the production of these cells, these permanent cells, these cells will not divide again, or it will not enter in any kind of cell cycle. So this three types of cells can be seen on the basis of their cell cycle. Okay. So before understanding the mitosis division in explanation, we should know that who actually discovered this mitosis division. The scientist Strasburger and scientist Walter Fleming, these two scientists actually discovered this mitosis divisions. In 1875, Strasburger, discovered the plant cell division, means the mitosis division on plant cell. Okay. In 1875, Strasburger discovered the cell division on plant, and 1879 another scientist who discovered the animal cell division, means or mitosis division in animal cells. We call the scientist is Walter Fleming. Discovered this. Okay, what he discovered? Animal cell division. And Fleming also named the name of mitosis. In 1882, Fleming also termed the name mitosis. Okay. So remember this names, scientist names, because it can become in your exam. Okay. So now we will understand in details explanation of mitosis division. Means already we understood the interphase. Okay, let me tell you one important thing in interphase. These cells, before the synthesize phase, before the entering of synthesize phase, it will get some check. Okay, it needs to check because maybe this cells is not able to produce or synthesize DNA. Maybe before due to the reasons of mutation or any kind of damage, this DNA or the genetic content may be damaged already. So what actually happened? Before the synthesize phase, here actually the checkpoint is present. Okay, means here some kind of enzymes or proteins which will check that this cell is able to produce new cells or not. So here the checkpoint is present, which we call the G1 checkpoint. Okay, which we call G1 checkpoint. After the checking of this point, means the cell is able to not able. After this checking, it will enter in S phase. Okay, in the S phase, it will produce or it will just double its DNA content. Okay. and after the double its DNA content, here in the position of before mitotic division, another checkpoint is present. And in this checkpoint, what actually it checks? It's just confirmed that this DNA is totally or ideally or perfectly synthesized, means the number of DNA is or the content of the genetic material is doubled perfectly. Whether it is perfectly doubled or not, that is checked here in this position, okay, which we call G2 checkpoint. Okay, G2 checkpoint. So after checking this point that this DNA is totally copied or not, if copied, then it will allow this cell to go for the next stage. If there is some error, then what actually happened? It will again copy the genetic content or repair the portion where actually the mistakes done. Okay. Then after repairing this phase, it will allow to enter in the next phase, which we call mitotic phase. So this is what actually happened in the case of this interphase. We already understood interphase. Now we will just understood, or the detailed explanation we understood in mitotic phase. What actually happened in mitotic phase. So before understand the mitotic phase, let me recap the portion which we already understood, that if I divide cell division, that how many types of cell division can be seen. So there are in our body cell, there are two types of cell, which is one is our somatic cell and another is our germ cell. Germ cell is divided by the meiosis division and somatic cell is divided by the mitosis division. And let me tell you that I already told you that there are one primitive type of cells present in the universe or in the earth, which we call the prokaryotic cell. Prokaryotic cells never go all these types of cell division, okay, never shows this kind of cell division. Their body or their cell organelles or the cell is very simple. They are actually unicellular organisms, okay, only one cell is present in their body, means their body is containing only one cell. So this cell will not go through this mitotic division or meiotic division. Not any division it will show. It will show different kind of division, which we call amitosis, okay, not mitosis, not meiosis. It will show different kind of cell division, which we call amitosis. Okay, this amitosis cell division is very simple method, okay. It is not complicated as mitosis division or meiosis division. It is very simple division, where just one cell will just increase its number, increase its genetic content and also the cell organelles which is present inside that cell, and it will just divide rapidly. It will just divide by producing the cell furrow, okay. By this type of cleavage it will produce and it will divide after few times whenever it will get mature. Okay, so this kind of division will not show prophase, metaphase, which will show can be seen in the mitotic division, okay. So that kind of division, which is very simple method, we call that is amitosis cell division. Now we will understand the mitotic cell division or detailed explanation of mitotic cell division, okay. One more interesting thing, let me tell you that in our body every minute 3 billion cells are producing, okay. 3 billion cells are every minute our body producing or dividing. Means every few days, after few days your body get all the organs are renewed, okay. New cells will produce. So in mitotic division, we already understood interphase, interphase. Now we will understand after interphase there is interphase is containing with G1, S and G2. So after the checkpoint of the G2 phase, the cell will enter in the mitotic division. Okay. In our body cell that will enter in mitotic division, and in our germ cell that will enter in the meiotic division. So in the case of mitotic division, all the things which will happen, that will be happen only in the nucleus. Okay. So first let me mitotic division can be divided on the 4 stages. Okay. First is prophase. All the 4 phases are prophase. First is prophase, second is metaphase, third is anaphase, and the fourth is telophase or telophase. So in the case of prophase means is the first phase. Okay, this is the longest phase actually. Longest phase, and in this phase what actually happened, the DNA or the chromosomes which is present inside the cell, normally in the cell, inside the cell, what character actually shows the cell in the normal time, not in the time of division. Okay, in the normal time, this nucleus means the nuclear membrane. Inside the nuclear membrane there is a chromosome which is scattered like this, unfolding. Okay, it's not coiled structure which we generally know. The chromosome structure is like this. This kind of structure it will not show this time. Okay, and nucleolus is present, and the centrosome is present here. Okay, centrosome is present here, and inside the centrosome there is a centriole. Centrosome, nuclear membrane. Why I'm showing you the nuclear membrane only? Because nuclear membrane is important here, because during all the phases sometimes nuclear membrane will disappear, and some phases nuclear membrane will reappear again, which we will understand. Just wait. So in the case of nuclear membrane, this is nuclear membrane, and nucleolus. Okay, and obviously other cell organelles, mitochondria, ribosome, all these things are present outside of the nucleus and inside the cell. So what actually happened in the first phase? In the case of prophase, this cells starts to enter in the prophase. So in the early prophase, what actually happened? This scattered DNA, or the scattered, not DNA actually, chromosomes. Okay, let me tell you that inside the chromosome, if you unfold the chromosome, it actually made up of DNA. Okay, so this unfolding chromosome will start to packaging. Okay, this unfolding chromosome will start to packaging, and it will start to thick in size. Okay, and it will produce like this type of structure. Normally this structure can be seen if you see through the microscope. The structure will be like this. This type of structure it will show. Okay, but during the time of prophase, it will start condensing. Okay, it will start packaging, and it will just this structure will package, and it will get a thick structure, which will like this kind of structure it will show during time of. Okay, these things are actually histone protein. Okay, these things are actually histone protein. These are histone proteins, and this is the DNA. Okay, so DNA is coiling in the histone proteins, and there are different subunits of histone proteins are present, which we will understand in the chromosome lecture. Okay, first understand what actually happened. This DNA will start packaging, and it will start, after packaging, it will show this kind of structure. Okay, this kind of this long strand of the chromosome will packaged, and it will condensed, and it will get shorter in size. Okay, so this is the longest phase. Prophase is also the longest phase, and it is also shortening phase of the DNA. Shortening means it will just get the DNA structure, or I mean the chromosome structure will get short. Okay, in this phase. DNA structure will get thick and short. So that cell during the time of prophase, how it actually looks, and this time, I mean the time of prophase, the centromere, or the not centromere, centrosome, actually the centrosome will also divide, and it will produce two centriole. Okay, it will just increase its number, and it will produce also two centriole, because two centriole are necessary. I will show you why it actually necessary. So it will start, and it will divide, and it will produce one more centriole. Okay, number of centriole get doubled, and this structure will be like the thickening structure. This kind of structure means that it's already get thick. Okay, and the membrane of the nuclear membrane, this also start to disappearing, and also nucleolus will start disappearing in this time. Okay, nucleolus and nuclear membrane will disappear in this phase, and this centriole will start to moving to the opposite side of the cell. Means two centriole will get in the opposite side of the cell. So this kind of structure it will show in prophase. And also one more interesting thing which will happen, this is actually happened in early prophase, and in the case in the time of mid prophase, what actually happened, this centriole will start moving here, and it will come here in this position, and it will start to producing astral. Okay, astral ray, this kind of ray it will start to produce, because from this kind of astral rays the spindle will also produce. I will show you what actually spindles. So after few times, what actually happened in the late prophase, okay, this kind of structure it will show in the mid prophase, and after the mid prophase there is a late prophase. In the time of late prophase, what actually happened, in the time of late prophase, this astral and the centriole will combine, or it will get attached in a thread-like structure like this. These are the spindle fiber. Okay, these are the spindle fiber, and nucleus, after packaging the genetic content, I mean the chromosome, after packaging it will looks like this kind of structure. Okay, this is one DNA, one chromosome. This is one chromosome. Means I am showing here only 4 chromosome. There is a 46 number of chromosome total present. Okay. So what actually happened? This spindle fiber, these are spindle fiber. This ray type of structure is the astral ray, and astral ray plus centriole we call aster. Okay, asters. And aster plus spindle fiber we call spindle only. If we combine aster plus spindle fiber, we call it a spindle. Okay, so this is happened in the case of late prophase. So after late prophase, what actually happened? It will enter in the metaphase. So during that time, what actually happened? This spindle fibers will go through in between the centromere region. Okay, in the DNA, in the chromosome. Suppose this is a structure of chromosome. This chromosome, here the centromere region is present. We call this region is centromere. And inside the centromere, the spindle fibers will go like this way. Okay, in the mid portion, which we call the kinetochore. This portion where the centromere or the inside in the centromere, the position where the spindle fiber is passing, that portion we call kinetochore. CHOR. Okay. So in the case of metaphase, what actually happened? This spindle fibers will passes through the kinetochore region, means the centromere region of the chromosome. So after prophase, during the time of metaphase, what actually happened? These spindle fibers will passes through the centromere region of chromosome. Like this kind of structure it will show. Okay. This type of structure it will show during the metaphase period or metaphase time. So let me tell you that these are the chromatin. Okay, this is the chromosome which is after the dividing, means this chromosome, these are the homologous chromosome we call. Okay, homologous chromosome means depending on the centromere region we number the chromosome. How many chromosomes are there? If anyone asks how many chromosomes are there, you need to count only this centromere region, not the arms. Okay, arms will increase during the time of cell division, during the time of synthesis, which I already explained. During the time of synthesis, first it will just containing one arm. Okay, this kind of structure it will show. But after the S phase, after the passes through S phase, it will synthesize. Means I already told you this DNA content will, or this genetic chromosome will just duplicate itself. Okay, and it will produce this kind of structure. The content of the chromosome is increased, but the number of the chromosome is fixed. There is also one chromosome is present here. So what actually happened? This is the metaphase. Here the chromosome is arranging in a pattern where the spindle fiber is passes through the centromere region, okay, of the chromosome. So after metaphase, it will enter in the phase which we call the anaphase. Okay, let me tell you that one interesting thing will happen in the this is happened in the early metaphase, but in the time of late metaphase, what actually happened, these chromosomes will arrange in the in this mid portion, okay, like this way. In the equator region here, in this region it will arranged, which we call the metaphasic plate, okay, or equatorial plate. In a line it will just arranged in this way, in one line, so which we call the equatorial plate or metaphase plate. So after metaphase, what actually happened, it will enter in the anaphase. Okay, so what actually happened in anaphase? So in anaphase, what actually happened in this phase, after metaphase, cell will enter in anaphase. In anaphase, what actually happened, these chromosomes will break down, means that inside the middle portion of the chromosome there is a centromere. This region will break, okay, this region will break and it will split two chromosome, I mean one chromosome will split into two chromosome, okay, and so means the kinetochore region, the chromosome will split in this portion and it will produce two new chromatid, and these chromatids actually are chromosomes which will produce the chromosome. So after splitting this portion, in this portion the chromosome will split, okay, and now in anaphase, the chromosomes which is already, first what actually happened, prophase, the chromosomes are start to arranging, and in metaphase chromosomes came in the mid portion, and after that chromosomes will start dividing, means in the mid portion it will just get split and it will come backward, means the chromosomes will get their position, okay, and because this in the mid portion the cell will divide, so they are coiling in the opposite side of the each portion. So in this case what actually happened in the anaphase, so in anaphase in the phase of this phase, these chromosomes will pull by the spindle fibers. These spindle fibers will pull the chromosomes in each side and it will get to the closer to the centromere, centriole region, okay. So this kind of structure it will show. I already told you before the cell division, this type of structure chromosomes actually have were, and during the synthesis phase, this chromosome will contain double its content. So this kind of structure it will show. In the prophase, actually the best during the metaphase, actually the best time to study the chromosomes, okay, because that time chromosomes are highly condensed and they are arranged in the mid portion. This kind of structure it will show, and after few times later this cell furrow will start, means the cleavage will start to forming the cleavage, because cytokinesis will also occur at this time, and after the anaphase, and this movement of the chromosome from opposite side or opposite pole of the cell, this movement we call the anaphase movement. Okay, after the anaphase, the phase which the cell will enter, which is called telophase. And during the telophase, what actually happened? All the things like cell membrane and not cell membrane, nuclear membrane and the nucleolus, which already disappeared, that will also again appeared. Okay, in this time, means during the time of, this is the structure which will show in the anaphase. And now during the telophase, in this phase, these spindle fibers will start to disappearing. These chromosomes will also start to uncoiling. This is the coiled chromosome, means that is totally folded. Okay, it's start to unfolding. Like this kind of structure it will show. Again this unfolding chromosome will appear, and it will cover with the nuclear membrane and nucleolus. Centriole will be outside of the nucleus and the nucleolus is present inside the nucleus. And here also same thing happened, means the nuclear membrane, inside the nuclear membrane chromosome is scattered position, structured, and nucleolus also reappear, and the cell membrane will start to formation in this position, and cell will divide after the telophase. Okay, so the telophase position, in the telophase this kind of structure it will show, and after the telophase this cell will already totally splitted and it will just divide. Okay, and it will produce two new cells. So this is what happened actually in the telophase. And after the telophase, if this cell is necessary to go to the next cell, I mean next cell cycle, then it will enter in the next cell cycle. Otherwise it will stay or it will get arrested in that position or in that situation. So this kind of structure it will show. After mitotic division, this is what actually we understand in mitotic division. Now let me tell you that what actually significance, why mitosis division is important. Because in our body we can get cut in any portion of our body, or we can get damaged in our body part or cell. During that time it needs to reappear again, or it needs to regenerate or repair our body part. To that repair, during the time of repair, this cell division, this type of cell division actually happened. Okay, means the mitosis cell division, which we understood, that kind of cell division happened in our body part. And also some wall lizard, you have seen that wall lizard, they just get cut their tail portion. So during the time of formation of new tail or regenerate the new tail, during that time also mitosis occurs. Due to the mitosis only they get their new tail. Okay, so and also in our body cell, the chromosome number, which is 46 number of chromosome, which is number of chromosome is continued. Okay, means the parent cell or the mother cell, which the also containing the 46 number of chromosome, and the daughter cell which produced just recently, that is also 46 number of chromosome. Okay, means the number of chromosome is also continued or maintained in this cell division. So this kind of things which is happened in the cell division of the mitosis. So this mitosis division is important for our body part, because even after the formation of zygote, what actually zygote, after the fusion of male gamete and female gamete, the zygote will produce. Okay, from mother the 23 number of chromosome will come, and the father 23 number of chromosome will come, and after the fusion the zygote, means the new cell will produce. and that cell will divide rapidly and it will produce a new born baby, means the new baby will produce. So during the time of zygote cell division, that cell division is happened by only the process of mitosis cell division. Okay, means mitosis cell division can be seen there also. So this is a very important type of cell division which occur in this case. So after mitosis cell division, we will understand the meiosis division on the next lecture. Okay, thank you.